speaker making her way to the stage. We have Paige Erpf, and she'll be talking about yeast. Let's keep the applause going.
Thanks, David. Hi everyone, I'm Paige. I'm a researcher from Macquarie University. Now my friends and family like to call me a synthetic biologist, a geneticist, a mad scientist. But do you know what I like to refer to myself as? A yeasty girl. Not because I'm harboring a yeast infection down here. Can't seem to get rid of the one between my toes. Just kidding. It's because for the last 8 years I have studied these fascinating organisms known as yeast. And tonight, I'm going to be telling you about all the cool things that yeast can do beyond giving you thrush or athlete's foot. So, many of you may not know this, but during the COVID lockdown, yeast was the backbone of keeping you all sane. It helped you get drunk, turned you all into exceptional bakers, and it kept you safe while you're all going at it like rabbits. And that's because it's been engineered to produce the Hep B vaccine. It's actually been engineered to produce lots of things, really. Take the oil industry, for example. It's been engineered to produce oils found in supplements, those found that are derived from fossil fuels, and in lube. Now, if you're thinking this is a new way to get a thrush, I'm not talking about personal lube, I'm talking about things like bike lube. You would have also started seeing you would have started seeing on the shelves meat-free and dairy-free products. Now, yeast is the technology or like the innovation behind a lot of these, and that's because it's been engineered to produce milk proteins or meat proteins that would otherwise come from an animal source. Now, arguably, this is more ethical. So why would we want to take this cool yeast and make it synthetic? Well, like anything good on this planet, we want to exploit the shit out of it. The problem is, we've kind of started to run out of ideas and what cool things yeast can do. But do you know who's really good at coming up with ideas? Evolution. Take, for example, the naked mole rat, the poodle moth, the blobfish. Like, look at this fucker. No one would have designed that in their right mind. But evolution did. The problem with evolution is that it's slow, and it keeps turning everything into crabs, eventually. We can't fix the crab problem, but we can fast forward evolution. And this was thought about 10 years ago by 2 researchers known as Jeff and John. And they came up with the notion of fast forwarding evolution in our favourite model of an organism known as yeast. And they thought, we can use yeast because it's easy to genetically manipulate, and if we want to turn evolution on and off, we can create a copy of it where we can do that. Super cool project. Do you know what they called it? Yeast 2.0. This is what I mean. No imagination. Version 2 of yeast. They had an opportunity to call it something really cool. Franken yeast, Yeastenstein, Yeastbot 5000, but no, they called it Yeast 2.0. So, if you want to know how to build a synthetic yeast and how to fast forward evolution, well, it's your lucky night, that's why I'm here, to tell you how to do these things. So, on to the first one, how to build a synthetic yeast. Well, when building any synthetic organism, you need to understand its genetic makeup. In the instance of yeast, we know that it's made up of 16 chromosomes. Those are made up of DNA. And we know that that DNA is made up of A, T, C's, and G's. Now, we call A, T, C's, and G's base pairs, and they're in a particular order in every single chromosome. So each chromosome has a different order. Now, this is the genetic code for the organism. And that genetic code is like the instruction manual that tells it how to be a cell. All right, and we have the technology to be able to determine exactly what that genetic code is. We also have the technology to be able to synthesize or print DNA. And with those 2 things together, we can create our own chromosome. But if we want to fast forward evolution, we need to put in these elements that can help us do that. And these are called LOXP sites. And we can insert them all throughout the chromosome. I will talk about how we do this a little bit later, so kind of save that in there. I want to talk about a downfall of this project. So the entire yeast genome, all 16 chromosomes, is 112 million base pairs long. We are only capable of printing around about 100 base pairs at a time, which means we would have to print 120,000 individual pieces of DNA, put it all together in the right order, which is a lot of fucking work if you ask me. But thankfully, we're not the only ones doing it. There's actually 11 groups around the world that are each doing their own chromosome. Macquarie University has got 2 chromosomes. So technically, the Yeast-Einstein project, Yeast 2.0, is a worldwide group project. It is going how you would think any group project is going. I'm not going to go into the details of it. But I will tell you one thing just to put in perspective. It has taken us 10 years to build 1 chromosome. Well, our lab has, anyways. Do you know how long it takes a yeast to replicate all 16 chromosomes? 90 minutes. So, if you thought we were playing God, you might want to reconsider because we're obviously not that good at it. Anyways, that's in a nutshell. I want to point out some poor fucker's going to have to put all of these chromosomes together. It's not us, thank God. It'll probably take them another 10 years. But that's how you build a synthetic yeast. So how do we fast forward evolution? Well, I'll give you some background on how we get evolution occurring naturally. Remember how I said we've got A, T, C's, and G's, they're all in a particular order. We can break those up into what we call genes. And for the rest of the talk, this is how I'm going to represent them. And we get mutations in genes. And when we get a mutation, we get a change in the genetic code, therefore a change in the instruction manual. And then we can get the organism doing something it wasn't doing before. This can be a good thing or a bad thing. So the different types of mutations we can get are deletions, where we get deletion of a gene, where we get duplications, where a gene gets duplicated, and we get translocations. Translocations are where we get a gene, and it's just decided it's had enough of its neighbor, and it just fucks the right off. It goes and inserts itself on another chromosome. Now, any one of these is going to cause quite a bit of a disturbance. And so we could get the cell doing something it wasn't doing before. And we can induce all of these things in a synthetic yeast. And that is where the LOXP sites come in. So LOXP is a specific sequence. It's the same all throughout. And it's kind of like a single or a beacon, which is recognized by Cree recombinase. Now this can get turned on, turned off, turned on, turned off, turned on, turned off. My partner probably wishes this is the control he had over my sex drive. But unlike me, when these guys get turned on, they cut off the LOXP sites. So, if you imagine DNA in a nice string, it pretty much goes around and just chops it all up and breaks it all apart. This is going to make the yeast cell pretty sad, because we're essentially butchering its insides. But like any living thing on this planet, it wants to survive. So it's a strong little guy. And it's going to start just stitching everything back together, grabbing DNA, putting it all back together, but it's going to do it in a rush, haphazardly, because we're essentially killing it. So we're going to get genes deleted, duplicated, removed, put somewhere else. And so it's obviously going to change. It's not going to be like it was before. Now I want to point out what was happening in one cell is going to be different to what was happening in another cell. And we can search for that to see which one's doing the really cool thing, so we can exploit the shit out of it. Anyways, I can go back to like normal. That's the end of my talk. Hope you all learned a little something about the language.
And that was Paige Erpf! Keep the applause going. We have our next speaker making her way to the stage. Keep the applause going for Elaine.